Regulation of digestion and absorption is really important to ensure that digested nutrients have enough time to be properly absorbed and that there's enough time to absorb the water from the digestive tract. It's also not efficient to be secreting lots of enzymes and salivary secretions and mucus if they're not necessary. So we want to avoid those wasteful processes. So digestive regulation can be divided into three phases: the cephalic phase, gastric phase, and intestinal phase. We'll look at the cephalic phase first, which begins with the anticipation of food. So the cephalic phase of digestion begins when the anticipation of food is integrated in the brain. So the sight of food or the smell of food is enough to start the digestive secretions moving. Salivary glands are stimulated by parasympathetic neurons. The salivary glands secrete water, enzymes, and mucus. Parasympathetic neurons also activate the enteric nervous system, which is located within the digestive tract itself. This has been called the little brain because it uses a lot of the same neurotransmitters that are used in the brain. Activation of the enteric nervous system increases the motility and secretions from the stomach and intestines, so increased contraction of smooth muscle, increased contraction of the gallbladder to release bile salts and bicarbonate into the small intestine, and also relaxation of the sphincter of Oddi. The sphincter of Oddi allows secretions from both the pancreas and the gallbladder to enter the small intestine. All of these activities signal the digestive tract to prepare for incoming food. The gastric phase of digestion is characterized by increased gastric and intestinal motility and secretions. The gastric phase is initiated either by increased parasympathetic activity, increased gastric stretch, so when food actually enters the stomach, increased concentrations of amino acids and fatty acids in the gastric chyme, and also a decrease in pH. Note that chyme got auto-corrected in this PowerPoint. It's actually spelled C-H-Y-M-E. Darn that autocorrect. So all of these stimuli above result in gastrin secretion by the G cells of the antrum of the stomach. So gastrin is a hormone. So gastrin has receptors in several places. One is the parietal cells of the stomach. When gastrin binds with receptors on the parietal cells, it results in increased secretion of hydrochloric acid, which, as we've learned before, converts pepsinogen to its active form, pepsin. Gastrin also has receptors on the chief cells of the stomach, and when gastrin binds with these cells, the chief cells secrete more pepsinogen into the stomach, which can then be converted into pepsin. There are also receptors for gastrin on the smooth muscle of the lower esophageal sphincter, which you'll recall is the ring of smooth muscle that separates the esophagus from the stomach. When gastrin binds with these receptors, it causes increased contraction of the lower esophageal sphincter so that the increased motility and secretions of the stomach don't get squeezed up into the esophagus, causing acid reflux. There are also receptors for gastrin on parts of the digestive tracts further downstream, so on the smooth muscle of the ileum and the colon. And when gastrin binds with these receptors, it increases contraction of this smooth muscle and moves digested material further along. So it allows more room to be prepared for incoming food. Following the gastric phase is the intestinal phase of digestion, which is characterized by hormones that actually reduce gastric motility and secretions. And these are hormones that are referred to as enterogastrones. Secretions are increased from the pancreas and gallbladder during this stage, and you can think of this as being characterized by the saying that you need to slow down to digest and absorb nutrients. The hormones that are secreted during this stage of digestion depend to some extent on what you eat. And as the acid chyme enters the small intestines, the composition of that chyme will influence what type of hormone is secreted. So for each of the hormones of the intestinal phase, we'll look at the stimulus for secretion, the source of the hormone, the name of the hormone, and the targets and tissue responses. So in all cases, the chyme that enters the small intestine is going to be acidic. So there's going to be increased acidity in the duodenum, which means we need more bicarbonate, which is the stimulus for the first hormone that we're going to talk about. This hormone that is secreted in response to increased acidity is called secretin, and it is produced by the duodenal cells. There are receptors in the exocrine cells of the pancreas for secretin, and the consequence of secretin binding with these receptors is increased pancreatic juice secretion. So this includes enzymes and in particular bicarbonate, which will help to neutralize that acidic chyme. The other role of secretin is as an enterogastrone, or a hormone that reduces the motility and secretions from the stomach or gastric phase. And so this reduces contraction of the smooth muscle of the stomach, which is what we mean by decreased gastric motility. And there are receptors on the chief and parietal cells so that there is reduced secretion of pepsinogen and acid. Also, another important thing that enterogastrones do is reduce the rate at which contents leave the stomach and move into the small intestine by causing contraction of the pyloric valve or pyloric sphincter. So this allows the acidic chyme from the stomach to dribble into the small intestine rather than to be dumped all at once, so that there's plenty of time to digest it. The next enterogastrone that we're going to talk about is one that is released if you have a lot of fatty and amino acids in your meal, which means you need bile salts to emulsify those fats. This hormone is called cholecystokinin, or CCK for short, and it is secreted by the jejunal cells, so a little bit further downstream than the duodenum. Cholecystokinin also has receptors in the exocrine cells of the pancreas, so there's increased pancreatic juice secretion, including bicarbonate. It also has the same enterogastrone effects as secretin, with receptors on the smooth muscle of the stomach and the chief and parietal cells, as well as the pyloric valve. In addition to those effects, cholecystokinin has receptors on the sphincter of Oddi and on the gallbladder. Since bile salts are needed to emulsify those fats, the contraction of the gallbladder causes a larger rush of bile salts to be released into the small intestine, and releasing the sphincter of Oddi increases the rate at which those secretions are released into the small intestine. The third hormone that we're going to talk about is secreted if you have a carbohydrate-rich meal, which is carbohydrates are digested into glucose, which means that you need insulin to allow that glucose to be moved into fat and muscle cells. The hormone is called GIP, or glucose-dependent insulinotropic peptide. An older name for it is gastric inhibitory peptide, which alludes to its enterogastrone effect, and it is also secreted from the duodenal cells. GIP has receptors on the beta cells of the pancreas, so when GIP is secreted, it increases the secretion of insulin into the blood to allow more glucose to be taken out of the blood. GIP also has an enterogastrone effect like the other two hormones, although it's through the release of another hormone called amylin, so it's a secondary effect. But just be aware that we'll lump it into the category of enterogastrone, just like cholecystokinin and secretin.